Yeah, I can see it. Yeah.
Okay, you can hear me. Okay, no problem. Okay,
Yeah.
so I will try to go a little bit fast because we have a video, right? And so as thank you very much for having me again this year. My name is Miguel Jaguari. I'm gonna give a brief intro of what I do in the lab. As Audrey already mentioned, I'm originally from Ecuador, South America, and I have been in Canada since 2011. But previously I live in the States since 2005. So I left my home country in early 2005. So I started as a marine biologist, and then shifted gears over the course of years into aquatic ecotoxicology, and then microbial ecology and molecular biology. So um, this is a little bit of a DNA test I did, just kind of for um, to learn more about my background. So this is just something that I add in my to my presentations, and I am mostly 70% of my DNA is indigenous, 20% roughly is from Spain, and the uh, remaining amount is typical from a person who has mixed races in their DNA. So this is just a picture of myself when I was a kid. I grew up in the countryside, and and as you can see, I have taken all the vitality and energy from my dad, and this is my dad. And I still talk to him on a regular basis. And but I just wanted to show you, only besides the journey as a professional or as a microbiologist, there is journeys that you take into in life, as a person. And thus, these are my siblings and I, my family and kids that I had at the time. I had two kids, now I have three. But then when I migrated into 2005 to the States, I didn't know any English. So I went into the University of California in Santa Cruz, of English as a foreign language. I was placed into a university, and that's the University of South Carolina, where I studied aquatic toxicology. But then I decided to change gears and study molecular biology, and that's my graduation here with my advisor. This was probably a week before I left the States. And at the time, I thought, okay, I was going to be back into my home country. But then within a month after I graduated from my doctoral studies, I got a job in UBC as a postdoc. So I had only 2 weeks of break, and then I moved to UBC in Vancouver, and I said, okay, I'm gonna settle uh, here with my whole family. But nobody told me that Vancouver was very expensive. So anyway, but then a position opened up here in the University of Manitoba, and then I apply and I got the job, which I consider, that was for me when I heard news that I have gotten the job. That was for me the my pursuit of happiness moment, because to get these type of positions are very difficult these days. But I I do think that this type of job or position, they are more and more difficult every day because of the competition. But also, for in my case, I have that passion, like most of the people that I work with, to teach the next generation of scientists. So in my case, the next generation of microbiologists. So and I study microbes, and microbes in this slide, what we have is that most of the we're basically human beings as such. We are composed mainly of microbes, compared to the human genomes, we only have 23,000 genes. But if you compare to the total number of genomes or genes that we have in our body, there are 1 million or more, over 1 million genes that are present in our body. And most of the time, these bacteria or these microbiomes is what keep us our human body working. And however, there is also microbes that some of them they are opportunistic pathogens, and they try to enter into our body. And that's the type of microbes that I kind of study. In this case, microbes that acquire genes of resistance. And the reason is because at some point, and it's estimated that in the year 2050, antibiotic death associated to antimicrobial resistance will be over 10 million, will cause 10 million deaths, surpassing other causes of death such as cancer and diabetes. So that's why I study this type of phenomenon that occurs in aquatic environments in particular. I do, I mean, I have been involved in clinical projects, but my main strength lies into the microbes that are found in the aquatic environment. So to study microbes, there have been two approaches that have been used. The one that relies on plates or media has been used for over a century. And there is another approach that was developed in the early 80s and is using a targeted PCR approach where a known sequence or a piece of little tiny pieces of DNA will amplify a gene that or a fragment of a gene that could be present in any given sample. Um, However, in the late 90s, a new approach was kind of developed, and that approach is called metagenomics. Metagenomics is a term coined by John Handelsman. John Handelsman is the or was the former scientific advisor for President Obama, but she coined this term, and metagenomics involves the study of DNA and as to study all the genes that could be part of that given sample. And and there is another type of approach that involves the insertion of those pieces into DNA into a vector, and that vector it will be analyzed for an expression or a production of a particular protein that confers resistance to a particular antibiotic, and that is called functional metagenomics. And the one that uses or targets all type of genes that are present in a sample, they are called sequence-based metagenomics. In my lab, we try to conduct both type of studies. And so we study the aquatic environment as a whole, meaning that we try to look for these microbes, mainly bacteria and viruses that could be present in aquatic sources. And then this source is passed to a process where the is treated. However, research has shown that some microbes can survive the process, especially in northern and remote communities. There could be a plenty of microbes that are present in these in the cisterns or in the water reservoirs. Once that this water is consumed, it passes through our guts and then it goes into wastewater, where there are different processes that involve to degrade the organic matter that is present in the fecal matter, and then to they are released those effluents into the aquatic environment where they can be used by other communities downstream, either for recreational purposes or also to for consumption. So those processes that occur in the aquatic environment can affect downstream communities. And also it's important to point out that the waterways here in Manitoba are all interconnected. So in the lab, we collect samples, and you will see that in the video what I'm talking about. So we collect samples from these aquatic environments, and then we use um, um, mechanical and serial filtration approach to kind of separate those microbial fractions, and then study, each of those fractions using molecular techniques. And the reason that we study these microbes is because certain microbes serve as a fingerprint of a particular environment. So we use these microbes in my lab as fingerprints of a particular environment. So, and these are the analysis, because besides the work in the lab, we also analyze those fingerprints, and they are servers or computer analysis that will help us to identify each of these, A, T, C, Gs. These are the sequences that we got from these type of samples. And further characterization is conducted not only at the taxonomic level, but also at the functional level using these state-of-the-art tools. The main goal of the project, or of my lab, is to find some of the drivers that could or the main parameters that could be driving some of the genes that we are finding in the wastewater treatment plants or drinking water systems. So we use statistical models to analyze these type of genetic information that comes out from these instruments and the aquatic environment per se. So, and this is just a general view of my lab, with the, these days, because of the pandemic, we have kind of to deal and do remote learning. But, I hope that in the next year or two, we can be back into our normal life before COVID-19. And finally, it's very important also to keep your life balance. And what I'm trying to say here, that family is very important besides publications, research, discovery. But it's something that I wanted to pass that message to you, because I do consider that you are the next generation of researchers. And thank you very much. And I think that's I think I have prepared a video for you. And if I will stop sharing right now, and if Audrey could please share the video with everybody, that will be great. And I will mute myself now. Thanks. Hi, everybody. My name is Miguel Jaguari. I have my lab here at the University of Manitoba, where I work with microbial fractions, meaning I work with microeukaryotes, bacteria, very tiny microorganisms called viruses. And probably you have heard lately a lot of viruses because we're dealing with this pandemic. And here I am so I can show you a little bit about what I do here in the lab with my students. Today is a little bit empty, the lab, as you could see, because it's a Sunday. However, I'm gonna give you a tour about my lab. What we do here is basically we study microbes in the aquatic environment. We study microbes in drinking water systems. We study microbes in source water, meaning the water that is used for drinking purposes. I also use molecular approaches or microbiology in general to study microbes
from wastewater treatment facilities, right? Because microbes go through different processes in our guts, then they reach these facilities where wastewater is treated so it can be discharged into the aquatic environment. However, I'm not denying the good role of wastewater treatment plants. However, we may need better treatments because from what we have seen, still there are microbes that can survive the process, and they can reach the environment and they can impact the aquatic microorganisms, or also small communities or rural communities that are that use this water or as a recreational water in the here in Manitoba. And so I'm gonna give you a little tour of what exactly we do. I hope you enjoy it. So thank you very much for your time. And again, I'll give you a tour, and I'm using my phone here, so you will have an overview of what I do here in the lab. But hope you enjoy. Thanks. So this is a tour of my lab that I share with another researcher here. But usually, once that we collect the samples, no matter what type of samples they are, like from the aquatic environment, they could be water, it could be sediments. We store the samples, or we keep the samples in these coolers when we collect them. And then what we do, we have plenty of ice packs, so the temperature doesn't fluctuate or change too much. And then we label each of these samples. The samples are wastewater, and also there is some solids. We have cake, and this is the byproduct that occurs or takes place in the wastewater treatment facilities, and then they will be used as a compost. But we're interested, in studying the microbes that can grow on plates. And these are some water samples that have been also collected in these facilities. They can also come from the river, like here. Last week we collected a sample from the environment, and it's water sample that has already been used, but we're looking for viruses here. And so this cold room keeps the samples, at 4 degrees, so it can be processed within 24 to 27 hours, so storage in the short term. And then after we collect these samples, then we are ready to process the samples. So the way that we process the samples, it depends on the type of sample, obviously. So for water samples, the samples go through different filters, because we want to separate them, or we want to enrich each of the samples. So we use these vacuum pumps, and then the samples are passed through filters of different sizes. Like for example, here we have 0.45 micron filters, we have 0.2, we also have 1.2 micron filters that we use. And we use some adapters, like the ones here in this bag. They have been already autoclaved, so meaning that they have been already cleaned, they are disinfected, and then they are ready to process, right? So then what we do is to pass the samples, in this case the water samples, through these filtration units. We place a filter there, and then we collect depending on what we want to do. Sometimes we just collect the fractions that are collected here, or the microbes that are collected in or trapped in this filter. Or depending if we're looking for viruses, we'll have to do a serial filtration using several of these filtration units. 1.2 micron filter, 0.45 and 0.2 micron filter, and then we work with the ultrafiltrate, or the water that has passed through that filter, so then we can extract the nucleic acids. And as you know, the nucleic acids in these microorganisms represent like a fingerprint for them, similar, to our DNA. So then we use this commercial approach to extract the nucleic acids. This kit is used for microeukaryotes or eukaryotes organisms like us. Microeukaryotes, because we are large eukaryotes. But anyway, so we use this type of kit so we can extract the DNA, and then the DNA goes through several processes that we kind of is mechanical and processes that basically will separate all the components to purify the DNA, and we use this microcentrifuge. Here, each of the students has a different workspace, so they can come and work with their computers, their laptops. So this is also a thermomixer where we heat up some of the components of the kit. And once that we have extracted the DNA, we quantify them using this instrument. This is a very precise instrument. Depending on if we want to measure DNA or RNA, we choose any of these assays and then just measure. And it may sound complicated, but it's actually it's a very simple approach. And we use standards to kind of calibrate the equipment, and then we insert one sample at a time, and then we quantify them, because it's very important for us to know the amount of DNA that is present in the samples. So these samples, after we have extracted, what we do is conduct quantification. And this is another space for a student, but this is our qPCR. And the qPCR, what it does is to quantify the genes or the genetic material. If you are looking for a particular gene that is specific for a virus, for a bacteria, so this is the instrument that you will end up using. So, and this is one of our previous runs from our student. And what we have here is basically several standards that are used. So with those standards is a known concentration, and then we place our samples, and then this calibration curve will tell us the amount based on these standards. It will tell us the exact amount of copies or bacteria in this case that we have in our samples. So that's a way to assess the quantity. And you probably have seen some of these assays lately when when people are talking about PCR for quantitation of SARS-CoV-2. So probably they conduct this run in one of these instruments, and this is the instrument that is connected to this computer, where you will quantify the signal of those gene copies that are present in that particular sample. In this case, it could be a specimen from the nose or from the oral nasal cavity, or it could, in our case, as we collect samples from the environment or from wastewaters, we want to estimate the total numbers of SARS-CoV-2 that are present in a community. So this takes place here. That's the way that we are currently quantifying or estimating the total number of cases that are present within a community. In the lab, it's very important to have equipment that will help you to conduct those extractions. So this is a very important piece of equipment that we have here in the lab, and that is called a biosafety cabinet. Biosafety cabinet, what it helps us is to protect the person working in the lab from being exposed to pathogens or that could be present in the samples that we are handling. So all the extraction takes place in these type of biosafety cabinets. So the researcher can work in a safe environment, and also the sample is also contained in this type of biosafety cabinet. So that's a very important piece of equipment, and it needs to be calibrated every year, so we kind of feel safe working here because there is an airflow that will avoid the samples or the microparticles to go into this area, right? So there are other pieces of equipment that we also use in the lab, like some of these microcentrifuges, some of these mixers or vortexes that what we'll do is to help homogenize a sample. You can see it here, just a demonstration. And what else? You already saw the instrument that is used to quantify DNA or RNA from the sample. Thermoshaker, and this basically it does is to shake the sample, and at the same time it can also set the temperature that you want to use to mix given a specimen. So it's a thermoshaker, the centrifuge, there is this plate spinners, the spin masters that you have here. What else? You have an idea of the pump. There is also a lot of glassware, as you can realize. There is a lot of consumables, so that are used on a regular basis in the lab. So so each student has an assigned project that it can be finished within 2 to 3 years if you are planning to do a master's degree, or if you are planning to do like a PhD or a doctoral degree, it can take from 4 to 6 years. So these are other parts of the lab. Obviously, you know, there is a microscope here if you want to do observations of microbes. So there is also incubators. If you want to grow a particular microbe, you can use a plate with agar. Agar is like a jelly type of material that contains certain nutrients where microbes will grow. Not all microbes, I have to point out that not all microbes grow under normal conditions, meaning that not all the microbes will grow on a plate. So that's why we use molecular approaches that can study a broader number of microbes in any given sample. So if the students are kind of need to conduct any weight out of the samples or the agar or any material that they need to prepare, they will use this scale, this analytical scale. There is a more sensitive scale right here. Again, on a microscope. And, you know, we also use fume hood. The fume hood is used for to conduct organical extraction or handling of chemicals. That could be risky also for the researcher to inhale the fumes. So everything takes place here in the fume hood. And that will keep you also safe to inhale any of those toxic gases. Let me see what else. So as you already saw, these are some of the containers that we use when we go to the field and we collect the water samples. The brown bottles are for wastewater as well as for sediments. For sediments, it's not we don't need a lot of samples per se. Less than 1 gram is good enough to extract bacteria. However, there are sediment samples that need to be kind of pre-processed with buffers. So this is one of the buffers that we use, so we can kind of homogenize the sample before extraction with any of these commercial kits. Um, Let me see, there is a set of pipettes, that we use to conduct all these different processes, extraction, addition of different aliquots into the sample. So because there is a lot of chemical reactions going on during an extraction. So that's why, sometimes if you are into science, if you are planning to be in science, it's very important to know also chemistry, some also some physics, and I will tell you why in a minute, because it's very important to to know some physics, because you need to be familiar with this type of instrument like the one right here, where you need to have certain speeds that you need to centrifuge or spin it down the samples. So that's why it's very important to keep in mind that. And we have also a record of all the people who use this centrifuge for processing the sample. So we know who was the last user, and if there is any particular issue with the centrifuge, which is a very expensive piece of equipment. So we already know who may have used it. But again, it's very important to balance correctly, because if this centrifuge is not properly balanced, there may be errors appearing or showing up here in the screen. So that's why it's very important to know about that. And also speed. Speed is also could be an issue, because if you don't set up the right speed, accidents may happen. So that's why we don't want that to happen. So that's why it's very important to know about those laws of physics. And again, everything, it needs to be also contained, like glassware. It needs to go into these containers. Also, the waste that we generate from these extractions also needs to go into these different buckets. They are assigned. So, and then you were wondering, right, what will happen once that you have this waste generated? Where does it go? So I will tell you in a minute. So all this waste goes to autoclave, and I will show you in a minute what I'm talking about. So this is an autoclave room. So there are many autoclaves here, and what an autoclave does, so if you are dealing with waste, it will go into this big container that it reaches a relatively high temperature, above 120, 121 to be precise, and then it goes through a 45 minutes time, and then all the material will be decontaminated here. So once all that waste is decontaminated in these autoclaves, we have to wait a few minutes, like 15 to 20 minutes, until it cools down. After the process has been finished and it cools down, then we can just use regular bags and then just place them here, so they can be disposed in a safe manner. So again, and also if you are use any type of container that needs, after you have washed and rinsed with plenty of water and soap, you need to kind of disinfect it, because some of this material is reusable, so it can go into these autoclaves to be decontaminated, so you can reuse it again. So there is a lot of trays that we use. There is also the bags, so if you are disposing, you can use these bags. If you are reusing, you can use this type of bags. That's the autoclave room that we use here in the Department of Microbiology, just to give you a little tour about it. Okay, so a few more comments. So always, I have my own library here, because After we do the extractions, we do the analysis. We also send the samples for sequencing, and sequencing, what we are doing is sending the samples to a center where they will identify, no, they will actually take those samples and pass it through an instrument, and then the instrument will the output will be sequences like have like 4 letters A, T, G, or C, and each microorganism has its unique signature. Then we use computer um, analysis and or aka bioinformatics, so we can analyze those sequences. And there is also servers nowadays with the internet. You can just compare those signatures with what is available in huge databases, so you can identify the microorganisms that you are dealing with. There is also more books about molecular biology. And let me see what else I can show you here. Um, okay, so there is also uh, instruments to determine presence or absence of certain material by using PCR. And in my lab, I use qPCR most of the time to quantify, not only to detect but also to quantify the genetic material that is present in some of these samples. So, um, I forgot to mention about the different reagents. So there is this is different reagents that we use in the lab, so we can enrich a particular pathogen or microbe that we of interest. These are reagents that can be kept at room temperature. There are other reagents that can need to go actually at 4 degrees, and they need to be like being a clean environment, so no other samples is present in this particular fridge. So everything is clean here in this uh, type of fridge. So we keep our reagents, and we have also samples that need, or actually, besides samples, there is reagents that need to be at -20, and these are some of the reagents that we keep them at -20 because otherwise they will degrade. And temperature plays a significant role of that, right? So we have heard about why it's very important to keep certain vaccines like the Pfizer vaccine at -70 at least. So that's one of the reasons. So we need to kind of uh, keep certain products at the temperature suggested by the manufacturer. I think that's it for today. And thank you very much for your time. I hope you enjoy. And I hope also, if you are coming on board in the Faculty of Science, to do your best. That's what, after all, that's what is all about, right? So give your best, give to you back to your community, and most importantly, be kind. Be kind with your peers. Be kind with people. So if this pandemic hasn't showed us uh, uh, kindness, I think we have failed as human beings, and I try to pass that message to my students as well as the people who work with me. So thank you very much for your time, and I hope you enjoy it. And look at this crazy dude. But I'm an average Joe. You will expect to have like a scientist with a crazy hair or crazy dudes. But that's pretty much it. And one final message. Let me see. So after all these studies, all the DNA, RNA analysis, computer analysis, computer analysis, the final goal for a researcher is to publish those results. Because if you don't publish, it's like you have never conducted the research. So that's another part that I wanted to say besides the message as a person, is to publish. If a researcher doesn't publish, that may represent the extinction of that researcher. So, always have good values. Sometimes it's very frustrating when the experiments don't work at the first or the second time, third time, or even many times. But don't get discouraged. That's the beauty of science and research. That you need to be consistent. You also need to record those mistakes so then that won't happen again. So that's why each of the students keep these records or these logs so they can uh, record how the experiment was conducted, and then they can learn some valuable lessons from that. But at the end, the goal of a good researcher is to publish good science. Good science that can be shared with your peers, and also your contributions towards this field of biology, and particularly microbiology. Thank you very much for your time, and hope you enjoy it. Bye bye.
Hello, everyone. My name is Parvin, and I think before I start, maybe Dr. Yuan could have a short introduction of herself.
Okay, good afternoon, everyone. My name is Chuyan Yuan, and I'm with the Department of Civil Engineering, Price Faculty of Science. And thank you very much to join us this talk. This shows that you are interested in studying the environment and care about the environment. This second, I would like to let everyone know that I feel extremely lucky and honored to work with all my graduate students. They are the driving force behind the research work carried in my research group. My graduate students, they are very talented, very creative, and hardworking. So I don't think I need to say too much here, and I would like to really to highlight their research. So I would like to pass on the microphone to Parvin. So Parvin, please take over from here.
Thank you. I'm going to share my screen. Okay. Can you see the screen? Okay. I'm going to get you a little bit familiar with environmental engineering. Environmental engineering is the application of scientific and engineering principles to improve and maintain the environment, to protect human health, protect nature's beneficial ecosystems, and improve environmental related enhancement of the quality of human. There are some disciplines in environmental engineering, and they continue to grow and mostly include water and wastewater treatment, air pollution control, groundwater and surface water flow and contaminant transport, solid and hazardous waste management, environmental impact assessment, and radioactive waste management. An environmental engineer needs to be equipped with some knowledge. One of the most important ones is chemistry. Environmental engineers need to be good in chemistry because pollutants are chemicals too. And to fight pollution, you should know the properties of chemicals. The other one is microbiology. The objects of this science in environmental engineering are the engineering systems of water, wastewater, solid waste, soil, and gas biotreatment, where biological methods are being used for treatment. The other one is soil mechanics. principles of soil mechanics as they relate to soil, contaminated groundwater, and waste materials, and to the design, construction, operation, and maintenance of structures such as waste landfills, tailings dams, and slurry walls are essential in geoenvironmental engineering. And fluid mechanics. Fluid mechanics helps us understand the behavior of fluid under various forces and at different atmospheric conditions. Fluid mechanics is present in the field of environmental engineering as the majority of environmental heat and pollutant transport processes are driven by fluid flows. And coding that has recently, especially recently, been widely used in environmental engineering. Computer science enables researchers and scientists to use large-scale data and investigate and analyze issues like climate change, water contamination, and waste management. An important aspect of environmental engineering and in general engineering is problem solving. Engineers generally think of themselves as problem solvers. Unlike scientists who examine the world around them to obtain the understanding of things, engineers are concerned with creating something new, something which is currently not in employment, while applying scientific findings. I'm going to explain this aspect with a very simple example of my own research. My research involves microbiology and soil mechanics. Microbiologists have found and studied a specific type of bacteria that is able to consume methane, which is a greenhouse gas, as their food and convert it to carbon dioxide. On the other hand, soil scientists have studied on different types of soils and their properties. So I use the knowledge of microbiology and soil science to design and construct a cover on a waste disposal site. In the cover, there is soil with the specific properties, and the soil contains natural methane oxidizing bacteria. So the methane coming from the waste could be used by the bacteria in the soil and be converted to carbon dioxide. In this way, by putting the science of microbiology and soil science in application, we could fight global warming. And this is how environmental engineering can work using science. Here, I was also asked Kadir, who is another PhD candidate in Dr. Yuan's research group. He has done some interesting research, and I think he's gonna share with you. Please, I'm gonna stop my screen share.
Thank you.
Okay. You can start. Thanks.
Okay. Thank you, Parveen. Hello, everyone. Today, I'm a PhD student in civil engineering, and Dr. Yuan is my advisor, and Dr. Viagwari is my co-advisor, and I work in both labs. Today, I will talk about my project about tracking COVID-19 with wastewater. As you know, most of the patients are symptomatic or asymptomatic. It is hard to detect the asymptomatic COVID-19 patients. And also it is hard to, and it is very expensive. It is very expensive. It is very expensive and time consuming to test all the individuals in a city to find the number of infected persons. What we know that the COVID-19 patients have coronavirus detected in their stool, and this stool is going to wastewater treatment plants, And as you can see, if we can detect it in the stool, then we can detect it in the wastewater too. So what we do, we collect wastewater samples from the wastewater treatment plants, and then we analyze it, and then we try to detect and try to estimate the concentration of coronavirus in wastewater. And accordingly, we inform the public health department that we actually, for example, we detect high concentration of coronavirus, which means that the number of cases are high. When the concentration is low, or when we don't detect anything in wastewater, it means that the concentration is low. So the public health department can inform public and can take some measurements to prevent the spread of coronavirus. So how do we process wastewater samples for the detection of coronavirus? Actually, Dr. Viagwari is he actually briefly mentioned about this. The viruses are generally much more smaller than bacteria, and we use the size separation method. Since their size is different, we use a filtration system. To separate viruses from the other particles, like bacteria and eukaryotes and maybe fungi and other large particles. So we know that the size of coronavirus is about it is smaller than 0.2 micrometer, while most of the particles, most of the bacteria are much more larger than 0.2. So we use a filter which has a 0.2 micrometer pore size, and when we filter samples with a vacuum pump, the liquid and also the viruses in it will pass through the filter, and the bacteria and other particles will stay on the filter. So we are processing this filtrate, which includes the viruses, to detect the coronaviruses. Our next step is to extract viral RNA from these samples, to extract the RNA of coronaviruses from these samples using some commercial extraction kits. And then, as Dr. Viagwari mentioned, we use qPCR to detect and quantify the coronaviruses in the sample. And by doing this, we can estimate the concentration of the coronavirus in wastewater. So I will show you some of our results. So we start sampling in July and we ended the sampling in December. And as you can see here, the bars are the concentration of coronavirus, and the line you see on the graph is the number of cases. As you can see, as the concentration of the coronaviruses increases, the number of cases are also increasing, and there is a positive correlation between them. And we collect these samples on different dates. And when we first detect the coronavirus in wastewater, it was September 13 when the number of daily cases were below 20, and when the total active cases were around 200. So actually, it's a good indicator that there will be an outbreak in the future. We know that the concentration is increasing, and based on this, we can inform the public health officials to take some measurements to prevent the spread of the infection. So, and based on this correlation, we can make some formulas to estimate the number of infection based on the concentration in wastewater, and then we can also use this tool as an early warning system. So we can predict a potential outbreak before it happens. That's how we use the wastewater as a tool to monitor and estimate the number of cases. Thank you for your listening.
Thank you, Kadir. I was thinking of playing a TED Talk from an environmental engineer before we move to lab tour, but I think maybe it's better to play the lab tour, and then if we had time, we could play the TED Talk later.
Hi everyone, and this room is U1236. It's our main chemical lab, and the whole lab is used for the data analysis and data acquisition. Very important lab. So in this room here, so basically we have thousands of high-tech instruments. So basically, yeah, here is the deionized water we produce the pure water for our analysis. Over there we have some instrument called HPLC, and this one have the TOC, total organic carbon. So basically, we measure the compounds in our water and wastewater samples. Again, here we have two big machines over there. We call the GC, which again you can make some very small amount of organic compounds. Back here we have a bunch of ovens, and so this one we're going to prepare our samples. And over there we have a flow injection analyzer. So that one is allow us to measure the phosphorus and the nitrogen in our samples. Okay, so let's move on to the next room. So this one is our undergraduate environmental teaching lab. So basically, all of the engineering undergraduate teaching activities will be performed in this room. Well, this room here, basically, as you can see, we have a whole bunch of equipment and instrument, and we have 10 working stations. So typically, the size of our class is about 70 to 75 students. So basically, every lab, we typically have 2 people work on one station, and we can house 20 students at one session. So from here, we have we can do the basic water treatment and the wastewater treatment research, and also demo some of the important the treatment unit process, and we can combine everything together into the whole unit. So this is a very nice lab, and I hope you guys are going to enjoy this room sometime soon.
Hello everyone. I'm sorry for the background sound of ventilation equipment in my lab. My research is to find a new material, an environmental friendly material to replace plastics. This is the mycelium-based biofilm, which is made from mycelium and waste. It performs a lot like plastics during its use, but are totally compostable at the end of its life. I'm working with different species of mycelium, such as Ganoderma species, Pleurotus species, and some trimming species. And also I'm working with different types of waste as substrates, such as some agricultural byproducts, some hampers, some coconut corn, and some textile waste. This mycelium-based biofilm has great chemical and physical properties. It could 100% degradable in our backyard or in our landfill, which is good for human and for our earth.
Hi, this is Tanvi. I am doing a PhD in the second year under the supervision of Dr. Jian and right now I am working with landfill leachate treatment, and I am doing the treatment with electroxidation process. So first of all, I will introduce and I will explain what is landfill leachate is. So actually, leachate is a kind of wastewater, and it is produced from the landfill municipal solid waste landfill site, and it is a mixture of wastewater, and it happens through precipitation like rainfall and snowfall through the solid waste and produced the landfill leachate, which is a highly contaminated wastewater. And it can create problem into the soil and also into the groundwater. So for this reason, this landfill leachate treatment is very much essential. So we collected this landfill leachate from the Brady Road Resource Management Facility in Manitoba, and now we are working with it. And this is the power supply. And through this, when we are turning it on, and electricity is coming out, and this is my electrode here. So from the electrode, they are producing charges and hydroxyl radicals, and these hydroxyl radicals is attacking the pollutant of the wastewater, and thus it is degrading the micropollutions and some organic and inorganics from the landfill leachate. So like this way, we are treating this wastewater, and this treated wastewater, this treated water, we can use in different purposes. For example, in recreational purposes in the park, and also we can use it in the toilet as a flush wastewater, as a flush water. Thank you.
All right, so my name is Carl Todd. I'm an MSc student here at University of Manitoba. What I'm working on right now is operating 2 MBBR reactors for a plant somewhere in the United States. We're treating wastewater from a meat packing facility. MBBR actually stands for moving bed biofilm reactors, and those are attached growth reactors where bacteria is actually attached to a plastic media, and we use that to treat the wastewater. So the goal of the research is to treat to remove organics and to remove ammonia from the wastewater before discharge. And yeah, that's the general gist of the research. There are some complex organics that we're trying to treat. It's kind of difficult at the moment, but with time we should get the system to work. And that's it.
Hello everyone, my name is Parvin. I'm doing my master's in civil environmental engineering under the supervision of Dr. Yuan. My research is on producing activated carbon from agricultural waste and investigating its efficiency for textile wastewater treatment. Activated carbon refers to carbon-rich material which, as an adsorbent for pollutant removal, has numerous applications, particularly in water and wastewater treatment industries, due to its high surface area, well-built pore structure, and favorable pore sizes. Renewable resources, which are cheap and widely available, such as agricultural wastes, with high carbon content, can be a promising option for activated carbon production. You can see examples of these agricultural wastes, such as rice hull, corn cob, oat hull, and coconut shell. The agricultural waste that I selected to work on is Moringa stenopetala seed husk. Moringa stenopetala is a tree indigenous to southern Ethiopia. Its seed oil is vastly consumed since it's beneficial to human beings. However, the seed husks are not edible and often thrown away, which can be converted to a valuable product such as activated carbon. There are four main steps involving in activated carbon production: sample processing, chemical activation, pyrolysis, and a final cleanup. So after all these steps, I have a high porous activated carbon which has the potential to trap contaminants. Then I use this activated carbon to see its potential for removing dyes from textile wastewater, because if dyes are not treated properly, can have negative impacts on the environment and human health due to their toxic nature. I selected two different dyes named Reactive Black 5 and Basic Blue 3. These dyes are adsorbed on the surface area of activated carbon. Through adsorption batch tests, I monitor the effect of different parameters on adsorption process, such as temperature and pH of the solution on the adsorption process, to find the optimum conditions in which the dye removal is maximum. So by applying activated carbon and providing the best condition, the final solution, as you can see, is entirely colorless. This means that the produced activated carbon has high adsorption capacity for dyes molecules.
My name is Sharmin Akhtar. I am a PhD student. My thesis title is Heat Transfer Model for Northern Landfill. What is landfill? Landfill is a solid waste management system where we can dump all kinds of waste such as municipal, commercial, industrial waste. So how heat is generated in the landfill? Heat is generated in the landfill due to biodegradation of solid waste with the presence of precipitation such as rainwater, snow, and so on, and also oxygen. Generally, heat is transferred to landfill surrounding by leachate through leakage of the landfill's liner and also heat conduction process. The objective of my research is to develop a conceptual model to simulate the heat transfer by using console software and to calibrate and validate the numerically simulated model with the field data. The impact of heat generation in the landfill are groundwater contamination and structural failures.
Hello everyone, my name is Parvin and I'm doing a PhD in civil engineering. My research is about mitigating methane emission from landfills through biological methods. Everybody has probably heard about global warming, and you might have seen the picture of a starving polar bear standing on a piece of melting sea ice. This is only one of the results of the global warming. The main cause of the global warming is greenhouse gas emission, and methane is an important greenhouse gas. The final destination of our organic waste is engineered dump sites called landfills. Once a landfill is filled to its maximum capacity, it is usually capped by clay cover and vegetation. So the organic waste inside the landfill start to decompose and produce methane. This methane emission can contribute to global warming. In my research, I am trying to reduce this methane emission through biological methods. So I constructed a 1-meter hole in a very big area of a landfill. Then I filled the hole by compost. Inside the compost, there are natural microbes that are able to consume methane as their food by capturing oxygen from the atmosphere and oxidizing methane. Then methane will be oxidized to carbon dioxide that has less global warming potential than methane. To see how much methane was consumed by microbes, I took gas samples from the biowindow regularly and measured the concentration of the gases in the laboratory. Then I did some laboratory tests, such as batch tests. In this picture, you can see saroon bottles that are filled by methane and a little bit of compost. Then I changed the temperature and moisture content and monitored the activity of the microbes. In this way, I could find the favorite moisture content and temperature for the activity and growth of microbes. Then I did column tests. I built the columns from PVC and all the joints and connections and hoses and flow meters were set up. I filled the columns by compost and fed the microbes by methane coming from the bottom of the column. And then I provided oxygen for them from the top of the columns. These columns could mimic the situation that BioCover had in the landfill. In this way, I tried to introduce more oxygen for the microbes living in the deeper layer of the compost and increase the productivity of the compost. So all these laboratory tests provided valuable parameters so that the bio window could work to its maximum capacity. Thank you for your consideration, and I hope it could be interesting for you. Thank you.
Thank you all for joining us today. Please follow us at LTS_U of M on Twitter, Instagram, or Facebook. If you haven't registered already, tomorrow is day 2 of EnviroTalks with Dr. Gail Davrin. She is a professor in the Department of Biological Sciences at the U of M. So feel free to register for that. You can also scan the QR code as well. And also, I just want to thank all of our guests, Dr. Jaguari and Dr. Wan and Parveen and Kadir for joining us today for EnviroTalks. We really appreciate it. Thank you for your time. And also, one more thing, we also have a podcast if any of you are interested. You can scan this QR code or look us up at Let's Talk Science's Declassified STEM Survival Guide. We're on Apple Podcasts, Spotify, Anchor, Google Podcasts, and more. We just finished our Farm Survival Guide series, which is out now. And next month, we'll have our first episode of the Med Survival Guide series, which is focused on medicine. So feel free to check us out there. So thank you everyone for joining EnviroTalks. We hope you enjoyed the first day of EnviroTalks, and we hope to have you all tomorrow.